हॅलो फ्रेंड्स, वेलकम. तर आता आपण 5th पार्ट बघणार आहोत, रेप्लिकेशन ऑफ द DNA. इट इज मोस्ट इम्पॉर्टंट पॉइंट 1 for all exams and very impressive one. The first one is dispersive replication of the DNA, second one is conservative replication of the DNA, and the third one is semi-conservative replication of the DNA. The third one is most important and successfully proved it, semi-conservative DNA replication of the DNA. We'll see the first dispersive method, dispersive replication of the DNA. We'll see here in dispersive replication, the old DNA molecule would break into several pieces. Each fragment would replicate and the old and new segments would combine randomly to yield progeny DNA molecules. Each progeny molecule would have both old and new segments along its length. You see, in dispersive replication, the each fragments would replicate and the old and new segments would recombine randomly and to yield progeny DNA molecules, and both old and new segments along its length. We'll see the mechanism. In this method, the two mother strands break at the several point, resulting several pieces of the DNA. Second point one is each piece replicates and pieces are united randomly. And the third one is thus two copies of the DNA molecules are formed from the single copy. And the last one is the new DNA molecules are hybrid, which have old and new DNA patches. We'll see here mechanism. After we'll see the diagrammatic representation of the dispersive replication of the DNA. This is the diagram of the dispersive method of the DNA. This is the parent and these are the first generation and this is the second generation, means first replication and the second replication. All are similar, and the diagrammatic representation show all the dispersive replication of the DNA. The drawback one of dispersive method, that is this method of the DNA replication is not accepted because such not universally accepted. Such replication would not proved experimentally because it does not prove with the experiment. This said on and we'll see this is the dispersive method. First one completed, and the second one is conservative replication of the DNA. We'll see this replication of the DNA states that according to the conservative scheme, the two newly synthesized strands, the following the replication of the DNA molecule would associate to form one double helix. One double helix will be formed, while the two old strands would remain together and as one double helix. Firstly formed one double helix, and the two old one forms one double helix. The mechanism of this conservative method. In this method, the two DNA molecules are formed. One molecule has both parental strands and other contains both newly synthesized strands. One molecule has both parental strands and other is newly formed. See the diagrammatic representation one. This is the diagrammatic representation of the conservative model. This is after first replication cycle. This is one parental and this is newly formed, and then second generation, this is parental and this is newly formed, and these are the similar after second recycling cycle. We remember the diagrammatic representation. We'll say all about the model or the method of the conservative replication. One also picture here. We'll see that this is the conservative, this is the parental DNA, this is the first replication, parental, same as parental DNA, and this is the second replication. This is also same for the parents, and this is the different newly formed. This is newly formed one. After second replication, two are newly formed, means parental. the drawback of this conservative method. This method is also not accepted because there is no experimental proof for this method. These two methods are not accepted because of the experimental no give the proper proof of experiment. Also we'll see the most important one, semi-conservative method. It is the semi-conservative method. According to the conservative scheme, the two newly synthesized strands following the replication of the DNA molecule would associate it to form one double helix, and while the two old strands remain together. This is conservative one, sorry, this is conservative one, not semi-conservative. See, this is conservative. Okay, so we move. This is semi-conservative mode of DNA replication was postulated by Watson and Crick along with the double helix model of the DNA. Watson and Crick postulated the semi-conservative method of the DNA replication. We will see the mechanism. This is the mechanism. We will see after. There are 9 steps or processes, mechanisms for the semi-conservative method. The first one is activation of the nucleotides. Second one is origin or initiation point. And third one is unwinding of the DNA strands. Fourth one is formation of the RNA primer. And the fifth is synthesis of the new strand. The sixth one is removal of the RNA primer. Seventh one is leading and the lagging strand. And the eighth one is union of the Okazaki fragments. Ninth one is formation of the daughter DNA molecules. We'll see first the diagrammatic representation. This is the diagrammatic representation. This is first see the mechanism. In mechanism, activation of the nucleotides. The first in mechanism, activation of the nucleotides. First one, the activation of the nucleotides. In the nucleus, there are 4 types of nucleotides in the form of their monophosphates, that is DMAMP, DTMP, DGMP and DCMP, and they are activated into their triphosphates via DATP, DTTP, DGTP and DCTP. This transformation is done with the help of ATP in the presence of enzyme phosphorylase. And we'll move. This is a adenine monophosphate. This is a adenosine triphosphate. Adenosine monophosphate and adenosine triphosphate. This is thymine, guanine, and this is cytosine. These are the monophosphates and these are the triphosphates of the nucleotides. And see the origin or initiation point. This is one important, origin and initiation point. Origin or initiation point. DNA replication starts at the specific point called as origin or initiation point. In eukaryotic cell, there are several such points. eukaryotic cell मध्ये खूप points असतात, initiation points. At the origin, DNA molecule breaks because of the formation of the incision, and incision is made up of due to activity of endonuclease enzyme. This is one most important enzyme, that is the endonuclease enzyme in the genetics. And the third step is unwinding of the DNA strands. You see, unwinding of the DNA strands. The unwinding of the DNA molecule, the true strands of the DNA starts unwinding with the help of unwinding protein, that is helicase enzyme. DNA molecule appears like inverted Y-shaped structure and replication fork. The two separated strands of the DNA are stabilized by helix destabilizing protein or SSBP, means single strand binding protein. This is enzyme is most important, helicase enzyme, and here helix destabilizing protein means single stranded binding protein. तर यामध्ये अनवाइंडिंग होतं DNA स्ट्रँडचं आणि ते वाय शेपसारखं, इन्व्हर्टेड वाय शेपसारखं दिसतं. दॅट इज द रेप्लिकेशन फोर्क असं म्हणतात त्याला. We'll move to the next, the formation of the RNA primer. This is inverted. 1 minute. Rotate it. Okay, that is visible now. See the formation of the RNA primer. The synthesis of the RNA primer is essential for the initiation of the DNA synthesis. This is most important sentence. DNAच्या सिंथेसिसला चालना देण्यासाठी RNA प्रायमरचं सिंथेसिस होणं खूप महत्त्वाचं आहे. नंतर RNA primer is synthesized by DNA template with the help of the special type of the RNA polymerase. This is also important enzyme, RNA polymerase. With the help of RNA polymerase, the DNA primers are synthesized by the DNA template. Move to the next, the synthesis of the new strand. Fifth process one. Here synthesis of the new strand. See the synthesis of DNA on RNA primer. The new DNA strand is synthesized on the DNA template with the help of DNA polymerase, and the DNA synthesis occurs in short pieces which are known as Okazaki fragments. This is the one new term, Okazaki fragments. We'll see in next process meaning of the Okazaki fragments. DNA polymerase enzyme in this process for the synthesis of DNA on RNA primer. The sixth one process, that is step, that is removal of the RNA primer. Removal of the RNA primer. Removal of the RNA primer. The RNA primer is degraded by the polymerase 1 by the DNA polymerase first. This enzyme catalyzes the synthesis of short DNA segments to replace the primer. Synthesis of short DNA segment ला catalyze करते हे enzyme, means the DNA polymerase enzyme, and for the replacing the primer, RNA primer. The newly synthesized segment is joined to the main DNA strand with the help of the DNA ligase enzyme. न्यूली सिंथेसाइज्ड सेगमेंट आहेत डीएनएचे, ते जोडण्यासाठी डीएनए लायगेज एन्झाईमचा वापर केला जातो. डीएनए लायगेज एन्झाईम, इम्पॉर्टंट वन. अँड द सेवंथ इज लीडिंग अँड लॅगिंग स्ट्रँड. सी हियर, लीडिंग अँड लॅगिंग स्ट्रँड. व्हॉट इज द मीनिंग ऑफ लीडिंग अँड लॅगिंग स्ट्रँड? ड्युरिंग द रेप्लिकेशन, द स्ट्रँड विच opens from 3' to 5' is called a leading strand. Where one trick here, 3 to 5 means lead होतं इथं, त्यामुळं लक्षात ठेवण्यासाठी बरं आहे. लीडिंग स्ट्रँड, लीडिंग टेम्पलेट 3 to 5 means lead होतं आहे. And its complementary strand is called a leading strand or the continuous strand. It is continuous one. It is constructed continuously at a faster rate मध्ये जाते. And the second one is other strand which opens from 5' to 3' is called lagging, means lag होते, कमी होत जाते, म्हणजे 5 पासून 3 पर्यंत म्हणजे कमी होते. याची एक ट्रिक आहे, लॅगिंग टेम्पलेट. अँड इट्स कॉम्प्लिमेंटरी स्ट्रँड इज कॉल्ड अ लॅगिंग स्ट्रँड. अँड इट इज कन्स्ट्रक्टेड डिसकंटिन्युअसली ॲट अ स्लोअर रेट. हे तुटक प्रमाणे तयार होते स्लोअर रेटमध्ये, अगदी हळू, आणि हे फास्टर रेटमध्ये. त्याचं डिफरन्स बिटवीन बघणार आहोत आपण. याचं बघूया डिफरन्स बिटवीन लीडिंग स्ट्रँड अँड लॅगिंग स्ट्रँड. दिस इज सिंथेसाइज्ड कंटिन्युअस मॅनर, दिस इज डिसकंटिन्युअस मॅनर. दिस सिंथेसाइज्ड टुवर्ड्स रेप्लिकेशन फोर्क अँड दिस सिंथेसाइज्ड अवे फ्रॉम रेप्लिकेशन फोर्क. Okazaki fragments are not formed in the leading strand. In lagging strand, the Okazaki fragments are formed. Enzyme ligase is not needed for the synthesis of leading strand, and here for lagging strand, enzyme ligase is needed for the joining small Okazaki fragments. इथं स्मॉल Okazaki fragments तयार होतात म्हणल्यावर लायगेज गरजेचं आहे. इथं तयारच होत नाही, त्यामुळे एन्झाईम लायगेजचा उपयोग काहीच नाही आहे इथं सिंथेसिस ऑफ लीडिंग स्ट्रँडमध्ये. नेक्स्ट पाहूया, द युनियन ऑफ Okazaki फ्रॅगमेंट्स. See the union of Okazaki fragments. Here the union of Okazaki fragments takes place with the help of the joining enzyme called as polynucleotide ligase. Important one, polynucleotide ligase. And the replication may occur either in a one direction or in both the directions from the point of origin. One direction or both direction मध्ये replication होत जाते. When the replication proceeds in both the direction, that is called as bidirectional replication. One new concept, that is bidirectional replication. The replication proceeds in both the direction, that is called as bidirectional replication. Simple one. And the last one is the formation of daughter DNA molecules. The C, formation of daughter DNA molecules. In this, the way for each old strand, a new complementary strand is constructed. Now one old and now one is new strand undergo coiling to form a two identical daughter DNA molecule at the end of the process. So this type of replication is called as semi-conservative replication, as the 50% part of the mother DNA molecule is retained and the other 50% part of the newly synthesized. Hence the process is referred as a semi-conservative replication. मदर DNA मॉलिक्यूलकडून 50% येते आणि 50% हे न्यूली सिंथेसाइज्ड होते, त्यामुळे याला सेमी कन्झर्वेटिव्ह असं नाव दिलं आहे. सो प्रोसेस खूप मोठी आहे पण खूप इम्पॉर्टंट आहे. दिस इज सी द लीडिंग स्ट्रँड इट इज मीन्स लीड होते फ्रॉम 3 to 5. This is the leading template 3 to 5, and here the let's see lagging template here, and this is the where is Okazaki fragments of the lagging strand. These are the smaller Okazaki fragments which grow from lagging means 5' to 3', 5' to 3'. These are the lagging strands. The small pieces are the Okazaki fragments, and this is the replicating fork. We'll see here, this is inverted Y shape one, this is the origin point, and this is the RNA primer. You see the process one important, simple but very vast to see. The most all the concepts are clear from this diagram. This is one lagging strand, this is leading strand, this is helicase for the cutting, and this is RNA primase, this is RNA primer, this is DNA ligase for joining, this is DNA polymerase for joining of the nucleotides, and these are the Okazaki fragments. This is the lagging strand. Topoisomerase, this is one new enzyme, one important concept, single. And this is also see the semi-conservative DNA replication. New daughter strand is formed here, and this is the polymerase enzyme, DNA polymerase. This is the DNA ligase, this is helicase for cutting the nucleotides. This is the replication fork at the incision point. This is the old parental strand, and these are the Okazaki fragments, and it is joined by using the DNA ligase. These are the single strand binding proteins. These are the proteins single stranded, important one. So we go to the next. So we stop here. Here difference between here the semi-conservative, conservative, semi-conservative, and dispersive. This is parental DNA one, this is the first replication, this is the second replication. Important is a semi-conservative. You will see the mother DNA molecule is 50%, and the newly formed one here 50%. It is called as semi-conservative. Conservative means the mother is all one is formed from mother, the other is newly synthesized. This is conservative. Dispersive means mixed, and here most important is semi-conservative. Also this is one picture, see, this is a semi-conservative, these are the parental, conservative, and dispersive model. We'll take a screenshot of these models which are useful to us for remember. So we'll see in the next module the significance of the DNA and some information about the RNA. So thank you friends. If you like the video, so share, subscribe. So thank you.